So this past year question is about ecological sampling. So let's look at the question. Grassland is an important breeding habitat for some birds, and these birds feed on plant material and invertebrates. Biodiversity of the habitat is maintained by domestic herbivores such as sheep, cows, and goats grazing on the growing plant material. So a group of students investigated the effect of grazing by domestic herbivores on the plant biodiversity of a grassland as measured by Simpson's Index of Diversity. They investigated two areas. One area was grazed by the herbivore. One area was grazed by the herbivore, and the other area was not grazed for many years because it was surrounded by a fence to keep out the herbivore. So there are two areas that they did the sampling to find out the biodiversity of the area, species diversity using Simpson's Index of Diversity. So question asked, state the data that the students would have collected from the grazed and ungrazed areas to calculate Simpson's Index of Diversity. So what is the data that you need in order to calculate Simpson's Index of Diversity? Meaning to say that even though you do not need to memorize the formula, from the past question I've seen, whatever statistical test, formula for statistical tests like chi-square test, t-test, to calculate standard deviation, standard error, to calculate Simpson's Index of Diversity, correlation, okay? So you actually need to know the components in the formula, and they can ask you in exam. So in this case here, based on the Simpson's Index of Diversity that you have studied before, okay, if you look at the equation here, the two variables that you need to use for calculations, small n, which stands for the number of individuals of a particular species, and the big N, which stands for the total number of organisms of all species. So therefore, you need to write these are the two variables, okay, in order for you to calculate the Simpson's Index of Diversity. So which means to say you need to know the components of a formula that you have learned in your syllabus. Okay. Next question. So describe a random method which the students could have used to collect the data needed to calculate the biodiversity of the plant species in two areas. And the description of method should be detailed enough for another person to follow. So this question is asking you, write down the detailed method. That means plan your experiment and write down the detailed method in order for you to obtain the data, which is the number of individual of each species and the total number of species, so that you can calculate the Simpson's Index of Biodiversity. So which means to say that you have to write all the methods involved in doing the sampling so that you can get accurate as well as reliable results. And therefore, this is a design experiment question, because you can see from here is 8 marks. Okay, so 8 marks means it's actually a design experiment question. So I've taught you before that every time when you got a design experiment question, always remember the subtopic. What are the subtopic that you need to have that when you write your answers based on the subtopic, you actually get the marks. So you want to actually start answering based on writing down the subtopic first. So in every design experiment, the first thing that you would like to talk about is actually the independent variable. So in this case here, the independent variable is actually so that means you have to take the sample from grazed and ungrazed grassland, right? So this is your independent variable. So there'll be 1 mark. Then after that, your dependent variable. Okay, your dependent variable means you want to take the data So what is it that you want to measure, right? Your dependent variable is what you want to measure. So what you want to measure is you want to measure, first, you need to be able to identify the species. Okay, I'm not going to write this yet because I want to write number 3 first, because number 3 and number 2 overlap. So number 3 is the detailed procedure. Now, because this is ecological sampling, so you want to write how do you go about to do your sampling. So which means to say that step number 1 is you need to have a map of the area at least. And on the map of the area, you draw the grid pattern. So draw the grid pattern on the map of the area that you want to sample. So after you draw the grid pattern, maybe, okay, last time you learned that you make sure that every box in your grid pattern will have the similar size as your quadrant, okay? So ensure that every box in the grid, okay, is similar size or represents the size of one quadrant. Okay. So after that, after you've drawn the grid pattern, you need to assign number to every box in the grid pattern. So therefore, you have to assign coordinate number or symbol, okay, to each box in the grid pattern. Right? So what do you mean by assign coordinate number? So this one, let's look at the meaning of coordinate. So this one here, right? Okay, I've enlarged this already. Let's go to here. Coordinate. Okay. So this is a very simple coordinate on the right side here. You see? So let's say if this is an area that you want to sample, then you draw the vertical lines and horizontal lines to make the grid pattern. Then every box in the grid pattern will represent the size of the quadrant that you want to sample. Then you assign the coordinate to each of the boxes here. So for example, in the x-axis, you use alphabet A to I. Then in the y-axis, you use a number 1 to 7. So therefore, this particular, for example, this particular quadrant here will have a coordinate of h and 4. So this is a simplified version of assigning coordinate to every box in your grid. But in actual case, if you use a map, every location in the map already got their own coordinates, right? So what is the coordinates? The coordinate is actually, in this case here, we can say it's a pair of values, which is x-axis value and y-axis value. Or in the actual map, the x-axis is the latitude, okay, versus the y-axis, the longitude. Okay, the latitude and the longitude. Longitude is like this, you know, become the x-axis actually. Latitude is like this. Okay, longitude and latitude. Okay, so what is longitude and what is latitude? So let's check so you know. Okay, look on the right side here. Look at the globe, okay, the Earth. So we actually assign the longitude and latitude, okay, based on the equator. You see the equator here is 0 degrees. And from equator all the way to North Pole and South Pole, we put 10 degrees all the way until North Pole is 90 degrees, right? So this is called the latitude. So latitude is in the y-axis, latitude, okay? So with reference to the equator is 0 degrees Celsius, right? So plus minus. Now, what about the x-axis value? So starting from the prime meridian line, right? Prime meridian line, which is from the North Pole that draw one vertical line all the way to South Pole. This is prime meridian line. From this point is 0 degrees. So from this point to the east is 10, 20, 30, 40, 50, all the way. Okay, so degrees, right? So this is the longitude, longitude, okay, longitude. So this is the longitude. So this x-axis value. And here is negative 10, 20, 30, 40, 50. So this is the coordinate on Earth. So therefore, when you see this one just now, it is actually indicating the exact location of different spots on Earth based on the longitude and the latitude. Okay, based on the longitude and the latitude. So this is called the coordinates. So every box on your grid pattern has got a coordinate there. So you assign a coordinate to your every box in the grid. And after that, you want to randomly choose which particular quadrant that you want to sample, and you want to choose randomly. So therefore, okay, so we continue with the answers. Therefore, in the detailed procedure, which is number 3, right? In the detailed procedure. Okay. So after you have drawn the grid, whatever points I've written just now, after drawn the grid and after assign the coordinates, so you have to choose the quadrant for sampling by use a method to generate random numbers for the coordinates. Okay. So this random number method, you can use a random table, random number table. Okay. Or you can use a random number generator. So this is actually programming that you can do it yourself, or you can actually buy this programming that, or maybe you can search for free random number generator online and generate the number. Okay, I think you definitely can get this one. Huh? Okay, so you need to indicate how you're going to get this random number. Okay, so and then you place your quadrant in the randomly selected coordinates. Okay, place your quadrants. If you look at the mark scheme, it is stated that students need to spell the quadrant correctly. So you need to spell the quadrant correctly in order to get 1 mark for point number 4 here. If the spelling is wrong, according to mark scheme, they will not give mark for this sentence. So therefore, you have to pay attention to ensure the spelling that you write is correct. Okay. So after you have set up your quadrant, you are ready to do your sampling ready. So you go to the quadrant, and what do you do there? So you need to identify the species. So after you identify the species, you count individual of each species in that quadrant, and then you calculate the total number of individuals of all species in that quadrant. So this is what you need to do. So you need to state this one. So detailed method. Okay. Now for identifying species, how do you identify the species? So there are many ways to identify the species. One of the easier way is you take a picture, upload to your computer, and you're using the Google to match this picture. Okay, you can match the picture, and then from there you find out, okay, what is the name of this particular species? Or there could be a website where you can match the picture for you to find out the name of the animals or the name of the plant. So how to do that? How to identify species? By matching the picture. Okay, so nowadays you can actually take a picture of a person and you go to Google and search for a picture that matches this person, and then find out who is this person, what do the person do. You can actually do that. Huh? So matching picture, or you can use a key. For example, a dichotomous key. So dichotomous key is an example here. Okay, So this is a dichotomous key here. For example, I think this is a better example here, right? So you want to find out the plant, division of plant. So you see, is this plant non-vascular or vascular? And if it's vascular, is it seedless or it is seed-bearing? If it's seed-bearing, is a seed naked or it's seed enclosed? So this example of dichotomous key. So you are dividing the structure into two different types, and then two different types and two different types to identify them. Like this one here, also same thing. Plants don't make seeds and make seeds. Make seeds got flower, no flower, got flower or not. Got no flower but can produce seed is conifer. Got flower and produce seed is sunflower, for example. Okay, so it is called dichotomous key. So you write that dichotomous key, the methods of identifying species, you get a mark. Because you want somebody else to carry experiment, you need to write very, very detailed. So standardize the procedure. Okay, so after the detailed procedure, remember you've got number one, you have mentioned already the independent variable, and number two, you have mentioned already the dependent variable is what you want to sample, what you want to measure. And number three is the detailed method. And of course, number four is standardized variable. So what are the variables here that you want to standardize? Okay, so the variable can be the size of your quadrat, the frame quadrat. Now in the mark scheme, they actually mentioned there are two types of quadrats here, either point quadrat or frame quadrat. Okay, now frame quadrat is actually made up of metal frame or plastic frame or flexible fabric frame. Okay, so it's a frame whereby inside it you have got strings to further divide it into smaller sections, just in case you want to measure percentage coverage. Then you can use those small boxes inside here to count the percentage cover. So there'll be actually strings inside your frame here, where you pull a string across your frame. So this is called your frame quadrat. Okay. So in all the frames here, all the strings inside the frame. Okay. And then another one is called the point quadrat. So the point quadrat is, one example is you've got a piece of wood, or a frame, sorry, it's a piece of wood. And to this piece of wood, you actually fix a metal onto it. So let's say you got 5 metal pointing downwards to the ground. Okay, 1, 2, 3, 4, 5 of evenly spaced metal, okay, onto the ground. So in the coordinates that you have selected as a quadrat, if you want to do point sampling, you will take this point quadrat. Okay, you'll take this one and you place it in that coordinate. And what you do is that whichever plant species that the needle point to, when you put this point quadrat down, you will count them. Okay, so you've got five points here. You have got 5 points. You've got 5 wire pointing downwards. So whatever plant species that it touches, you will start to count them. So you can see what is the difference between the frame quadrat and the point quadrat. Okay, so this is the frame quadrat and this is the point quadrat. So which one do you think will give you a more accurate measurement? So the frame quadrat will give you a more accurate measurement because it's a bigger area for you to sample. Okay, so frame quadrat will give you a more accurate measurement. So point quadrat is less accurate. But why is it people want to do a point quadrat? It could be less time consuming. Okay, so that means when you choose the method of sampling, You will choose on the accuracy that you want, as well as how much time you are willing to spend or that you have to do the sampling. So let's say, okay, you don't have much time, but you want to take a rough sampling of the whole area. Maybe you want to choose point quadrat. So very quickly, you can sample a huge area for a rough estimation. But if you want more accurate, you need to spend more time and you use frame quadrat. So that's why you got frame quadrat as well as point quadrat. Okay. So therefore, you can say, okay, in the detailed method, you can say that in the coordinate as is selected, you can use a frame quadrat or point quadrat to do the sampling. Okay. So standardized variable, you standardize the size of the frame quadrat that you want to sample. Another thing is you actually want to standardize your number of quadrat or the number of samples that you're going to take in this area. Okay. But if you see in the mark scheme that they also say that you need to standardize the number of plots. So what do you mean by standardizing the number of plots in that area? So there are a few terms here you need to understand. What is the number of plots? So let me bring up the picture for the number of plots. Okay. So if you see the diagram on the right hand side here, I'm not going to remove the words because I need it afterwards. Okay. Okay, where's my diagram? Hang on. Here. Okay. So let's say this green area, right, is the forest area that you want to find out the biodiversity or species diversity of this area. So this the whole area is the green color area. It's a forest. Okay. So what you can see is that for B, grid pattern has been drawn onto the map for this area. And then you can see that for so this grid pattern has divided this area into different plots. That means to say that this plot, right, this plot here, okay, this particular plot here, all right, it is bigger than the size of your quadrat, and your quadrat is the red color box there. So because this area is very huge, you don't want to be drawing so many lines until you can divide the entire area into the size of your quadrat, because this area is so big, right? So therefore, instead of drawing the grid pattern to align with the size of your quadrat, you want to draw the grid pattern to divide them further into plots, so different plots. And then you randomly choose which are the plot that you want to sample further. So you can use random number to choose the plot. So after you've chosen the plot, you can go to the plot and do your sampling. So you can put your quadrat in the plot. So what you can see here is that for this particular example, after they have divided into plot in sample B, to the center of each plot, they put the quadrat and they do a systematic sampling. So you can see this is actually a systematic sampling. And this is also, this is a systematic sampling. This for every plot in the center of plot, I put my quadrat. So this is systematic sampling. All right. In example C, where's my example C? This example C, yeah, okay. For every plot that I have, I do my sampling. So I put a quadrat there, but the quadrat within the plot is further selected by random sampling. So that means I can further divide the plot into a grid line. So this area got their grid line, and every plot also got their grid line. And based on the grid line, right, I randomly choose where I want to place my quadrat. So every plot, I've got one quadrat. So you can see every plot, the quadrat location is different one. Okay. Or for D is I also draw the grid line for my area to divide them into different plots. And in each plot, I set up 4 quadrats that are stick together to each other. And I choose the location of the quadrat by random sampling. So in this case, I don't sample every plot already. You see, every plot there, I randomly choose the plot. That means I have randomly chosen the plot. And then for the plot I've chosen, I randomly choose again where I want to put my quadrat. Okay, so all these sampling methods are okay as long as you write very clearly in your methods and procedures. How do you go about doing the sampling? So you need to standardize your sampling method. So all these methods here are actually standardized method, which you need to write down. Okay, so these are the standardized methods of sampling. So let's go back to the question and continue. Right, so number of plots in the area that you want to sample is standardized, either all of them or randomly choose them. Okay, so this is called standardizing. So after that, we go to number 5 of your methods. So number 5 is actually methods in order to obtain reliable or accurate results. Okay, usually you know that in order to obtain reliable results, you have to have replicates, right? So you need to repeat the experiment, repeat the sampling in different plots. So you have divided the area into different plots. You have to repeat your sampling in different plots. So in plot 1, you sample, you take 10 quadrats to sample. Then in the next plot, you also take 10 quadrats to sample, or you sample 10 quadrats. So repeat sampling in different plots. Okay. In the same area. In the same area that you want to sample. So the entire area. Okay. Repeat it. Okay. There's one more thing you want to standardize here. Now you are sampling the plants, right? Okay, you're sampling the plants of the 2 areas, right? So you know that the presence of plants and the condition of plants will change with season. So one of the standardized variable is you may want to sample in the same week or same month, okay? Because in a country with 4 seasons, right? Spring, summer, autumn, and winter, temperature will change, rainfall will change, sunlight will change, sunlight availability will change. So that will actually affect the density of the grass in that area. So you need to make sure that you standardize the timing of sampling, the range of time. Even like in Malaysia, even we don't have the 4 seasons, we have got dry seasons, monsoon seasons. So if you're talking about grassland, right? Usually when it's very dry for a long time, the grass cannot grow, so they're gone, okay? And then when it starts to rain again, the grass will start to grow and spread, and it becomes green again. So therefore, you need to standardize the range of period of time or day that you are doing the sampling that is in the same season. So that is standardizing the time that you want to sample. Then of course, number 6 in subtopic for design experiment is safety. Okay, so you want to write the word safety first in your answer. So write like what I've written here. Write your subtopic, and then under the subtopic, write the points for the subtopic. So if you write like this, right, you see that for every subtopic you write one answer, you already get 6 answers. 6 answers is 6 out of 8. And then you got 2 more to write, maybe 2 more for standardized variables, right? Then you get the full marks already. So I highly encourage you to write your answers for any design experiment question in this manner, right? So safety, so you're going to the grassland to do sampling. Your safety precaution is very important. So one of the safety precaution is you want to ensure that you stick to the group to prevent getting lost. So you say what is the method of safety precaution means you stick to the group. Why? Because you want to prevent yourself from getting lost. Okay. And then number 2, you'll be exposed to all the plants and pollens in the air, and also maybe the mosquitoes and insects. So you want to protect yourself, right? Protection. So protection is a very general term. You will not get marked. So you tell me what are the gears you're going to wear. So maybe you want to wear mask. So wear mask, okay, to prevent pollen, to prevent breathing in pollen, which may cause allergy or even aggravate asthma. Asthma, okay? So that is if you wear mask. What about protection? I have to wear long sleeves and pants, long pant. Okay, so this one, because you're going to the grassland, you want to cover your skin away from mosquitoes. So you want to wear long sleeves and long pants to prevent insect bites. Okay, so this is called crystal clear answer. Please write very clearly how you're going to protect yourself, for what purpose you protect yourself. Okay, you want to wear gloves. Okay, you want to wear gloves because you might be touching the plant and it may cause allergy to your skin. So prevent skin allergy. Okay, so that's for protection, exactly what you do. Anything else for safety? I think, yeah, you need to wear shoes. Don't wear slippers. You want to cover your feet, okay? Wear shoes. Wear shoes because some area that is wet may have got leeches. Prevent leeches, okay, from crawling to your feet and start sucking blood. Sometimes you wear shoes is also not enough. Wear shoes also, yeah, some of the plants got a lot of spines, like needle-like spines that may poke your skin, right? The grasses. So you want to wear shoes to protect your feet. Whereas for leeches, they are able to actually they'll suck onto your shoes and they start to crawl on your shoes and go up your leg one, okay? So even you wear shoes also might not protect yourself. So they said that you need to wear socks, long socks, and you use rubber band or use string to tie your socks and your pants together to prevent the leeches from going in. Yeah, so that is for preventing leeches. So all these things you can write for safety precaution is 1 mark. Altogether 8 marks for this section, okay? Right, next question. So next question is very straightforward. So the question asks student, the hypothesis of the student is the mean height of the plant. So this is your dependent variable you want to measure. It is greater in the ungrazed grassland versus the grazed grassland. So it's very clearly showing you that there are two different conditions that you want to sample. So your independent variable here is the grassland that is grazed and ungrazed. Okay, grazed and ungrazed grassland is the independent variable. And your dependent variable is the mean height of the plant. Okay, exactly what do you want to measure? Height of the plant is also acceptable. Mean height is also acceptable. Okay, so this is the data obtained from students. So the student took 10 samples, right? Sample number 10 for these two areas and measured the height plant in mm. So it's very interesting that the students are asked to actually calculate mode and median in this question and write your answers here. So which means to say that you actually need to know how to calculate mode and median, right? And if you do not know, if you can actually check, okay, this value here and this value here against the data, how do they get to the answer? So that's why based on this question, it actually tells you that everything that is listed in your syllabus that you need to know, you need to know. Okay, you cannot say ignore and you do not know. You make sure that you know because you want to score full marks, okay? So therefore, that brings you to the notes that I've given to you. This note is very good. You need to actually, please refer to this note and make sure that everything in this notes, everything in this notes here, right? From planning, experiment to analysis, evaluation, conclusion, everything here is really, really good. Like for example here, mode, mean, and median right here. Can you see here? Mode, mean, and median that you need to know. And also this part here, make sure that your notes that you have is the latest version, right? The latest version, I've actually illustrated how to find out interquartile range. I've got this diagram here on the right side. Make sure you've got this diagram here or else yours is an old version. So you have to ensure that you print out the new version or you download the new version, all right? So even like this one also here, where is the range, where is the median, where's the interquartile range, okay? What is the distribution like? So this is the latest version that I want you to have. Even like this graph is only found in the latest version. Okay, make sure you have the latest version with you for your notes. Right, So now here they ask you to calculate the SM value. So SM is actually the standard error. So they've given you the standard deviation, right? Standard deviation is actually the spread of the data around the mean. So this one you have to know. Spread of the data, individual data, right, around the mean. Okay, whereas standard error is actually the spread of the mean around the mean. Okay, the spread of the mean around the mean. So what do you mean by spread of the mean around the mean? Let's take opportunity to do a revision here. So let's say this is a group of students in Taylor's College, and for an exam paper, let's say trial exam paper, right? Let's say for a trial exam paper, we do not know whether this paper is too easy or too difficult or it's just nice. So therefore, we will actually select sample students from, let's say these are all the A-level students that will want to sit for the trial exam. So we will maybe randomly choose a group of students here, okay, to do the questions, and do not know that these questions are the trial exam questions. And then from their results, we will find out what is the distribution of their marks and what is the mean marks that they get. Okay, so that's called the mean value. Then after that, from this mean value that I get from this particular sample, let's say I got a mean value, mean from this sample, mean from sample 1. Now from this mean sample 1, I can use it to predict. So for the entire population here, what would their mean be located? So what kind of mean they will get? So that I have to use this standard error to calculate. That means from the sample mean, I want to predict what is the population mean. So I use a standard error. Or from the sample mean, I want to predict what is another sample mean. So that means if I go to another class and test them, what will I expect the mean value of this paper to be for this particular sample of this population? So therefore, it is predicting the mean, where is the spread of this mean from another mean that you have obtained from this population. Okay. So they ask you to calculate standard error, so which is very easy. Just take the standard deviation and then chuck into here, and number of n is equals to 10 just now from the sample, right? So this is very straightforward. So let's answer the question here. Okay, so based on the answer given, let's assume that you have calculated the value. Standard error for the grace area is 9.33, and the standard area for ungrazed area is 4.97. Now remember, they actually indicate to the student how many number of significant figure they want. See here, give your answers to the 3 significant figures. So therefore you give your answer to 3 significant figures. So when you're asked to do calculation like this, right, usually don't round up first until the final calculation. So after the final calculation, you round up based on your original data. What is the total number of significant figures, or what is the total number of decimal place? So if your calculation involve multiplication or division, then you have to follow the number of significant figures of your data. If your calculation only involve plus or minus, then your final reading calculated value must follow the number of decimal place of your data. Your data with the least number of decimal place, you have to follow that one. So it all depends whether you have done multiplication and division or not, or plus or minus, then which format to follow for your number of significant figures or number of decimal place. Okay, so let's go back. I've deleted everything. Okay. So here is a very important concept that they ask you to calculate. So standard error is used to calculate 95% confidence interval, CI, 95% confidence interval. And the CI is given as mean plus minus 2SM. So you have actually learned this one already, right? The confidence interval with 95% confidence is mean plus minus 2SM. So they have given you this value for grace area is 548.3 mm and 585.7 mm. So that means to say that when they ask you to calculate, right, your result also must give to a 1 decimal place. So this is the tips when you're not sure how much to write, you follow whatever that's given there. So this is actually a tip for you. So the answer for the confidence interval for ungrazed area, okay, is equals to 860.1 to mm to 879.9 mm. Okay. So after having obtained this information, the question below further asks you this one. State what information is gained by calculating the confidence interval. What information is gained by calculating the confidence interval, right? So this one you have to go back to your theory. What does the confidence interval tells you, right? So first thing is, now this one you're familiar, right? Okay. What do you mean by this is whenever you have got this confidence interval that been calculated, right here, this value and this value that you've calculated, it is telling you that if you go back to the same population and take the trial paper and ask any sample from here, you randomly choose a few students from this group to do the paper, you will have 95% chances or 95% of the time that the mean value of this small sample will lie within the 95% confidence interval that you have calculated. So that's the first meaning. So 95% of the chance, the mean value of the sample that you have obtained from the same population will lie within this confidence interval. Okay? So that means you've got 95% confidence about this mean value will be correct, will be within this range. So this is the first marking point. Okay? Then the next thing you can see here is it shows reliability of the mean value. Okay, for example, standard deviation shows the reliability of the data. That means how close are the data distributed around the mean. This is how close the mean value of any samples you take will be close to the first mean that you've calculated or will be close to the population mean that you've calculated. So it shows the reliability of your mean value of your sample, how close it is to your first mean or how close is it to your population mean. So reliability. So that goes back to your notes. Okay, this is your notes here. So the notes for standard, sorry, notes for standard error. Okay, here. I don't want to remove my writing because I need it later on. So I want you to read that the standard error stands for the spread or the closeness or the reliability or the accuracy of your sample mean away from the actual population mean or away from other sample mean. Okay, so that's called reliability. So this is the information that you can get. Second information. Okay, so third information you can get is you compare between these two. So everything you have learned about the confidence interval. Now this confidence interval here, right, is actually the error bar, right? And you have learned that error bar show the spread of the mean. So when you plot the graph and you draw them as error bar, you'll find that this range here do not overlap with this range. This range here does not overlap with another range. So when they do not overlap, what does it mean? It means that the differences in the mean height of the plant in the grazed and ungrazed area is significant. Okay, so that means to say you need to know your basic very well so that you can answer this question. This question is 2 marks. So you need to write more in order to score the marks. All right. Okay, the next part is mark release capture method. So the question asks the student, state 2 precautions that the student should have taken to ensure that the results they obtained were valid. So that means all the precautions that you can think of for capture recapture technique. Okay. So let's list them out. What are all of them? Okay, number 1. Number 1 is the capture recapture technique will only be successful if you are doing sampling in a large area. Okay, in a large area, so that whatever organism that you have sampled and marked, and when you want to release them, you allow them to move around the area to mix with other organisms. The success of this method depends on the ability of the organism to mix with each other. Okay, so let's say you've got a fish tank at home, you don't have to do the sampling because one glance already can see everything. So you want a large area to allow mixing to happen. Okay, then you need to allow enough time for marked organism to mix with unmarked organisms. Okay, so the success of the procedure depends on the ability to mix. And then the next one is about the marking. When you do the marking, you must make sure that it is not toxic to the organism. Okay, you must make sure that it will not be not washed by rain or being rubbed away by the animal. Okay. You also must make sure that the marking will not make the organism more visible, the predator. Okay, so you must not affect the ability to survive by marking them. Okay. Okay, allow enough time. Now you allow enough time for them to mix, but cannot be too long until this organism disappear from that area, until they migrate away. Okay, or they've changed in stages of their life cycle. Now, change in stages of life cycle means what? For example, you want to sample tadpole. So you know tadpole, right? They've got a certain time where they will actually metamorphosize into frog. So you cannot leave, after you mark them, you put them back to the water. After that, after 3 weeks, you go back, they're no longer there, right? All the marked tadpoles have become frogs, and then none of them are marked already. All right, so that's the meaning of change in stages of life cycle. Okay, and of course, marking also, you don't want the marking to hinder them to be able to move around or to swim or to fly. So it must not hinder them and make them unable to survive better. Okay. So last question. The population of an invertebrate that feeds on the seeds was estimated in both the grazed and ungrazed area. Predict which area will have the greatest population and give a reason for your choice. So this invertebrate feed on the seeds. So which area between these two areas, which is grazed and ungrazed area, do you think you'll be able to find more of this invertebrate? So when you want to have got more invertebrate, you must have got more seeds, right? You must have got more seeds. When you want to have got more seeds, you must have got more plant. So where's the place with more plant is the ungrazed area. So your choice is, of course, ungrazed area. The reason is because they got more plants. Okay, so we have completed the discussion for ecological sampling.